Okay, so let's get started. I'd like to just quickly introduce Ophelie, who's one of the other new speakers this week. Ophelie is from Montpellier, and she has again studied niche evolution and range expansion and done a lot of modeling work with this for years now. And so again, she's going to tell us about niche and geographical ranges, how they evolve, how dispersal evolves, and so on in the 5 lectures that she has this week. Welcome.
Thank you. Okay, so I work in Montpellier, in southern France, and in some institute which is called Institut des Sciences de l'Évolution. And during this week, I'd like to talk generally about how population genetics impacts the dynamics and demography of populations, and on the way back, how the dynamics and demography of population is changing. Change is affecting change in gene frequencies and selection in those populations. And I will particularly focus on the effect of, okay. I don't have a phone. Okay. So I will particularly focus on dispersal and also spatial heterogeneity and spatial structure, because maybe it's something that you heard less of until now during this course. So that's the general framework, and through this kind of general interaction, I will illustrate different specific questions. So today, I will talk about the evolution of species niche, species ecological niche. So this concept was initially a purely ecological concept, describing so the set, the definition by Hutchinson in the late 50s was that this niche would describe the set of all environmental conditions where a species can persist. So it means that, for instance, it would describe the temperature, the quantity of nutrients, or the other aspect of the environment, such as that if you plant some plant there, it would grow and survive and produce seeds such that it would maintain a viable population in this environment, if you exclude other type of interventions. So basically, the fundamental niche is where a population of that species would be able to grow and have a positive growth rate defined by, I think you probably saw that with Maria during my lecture, which is related also to the natural logarithm of the geometric population growth rate. So then immediately there was another definition of the niche, which is the realized niche, because the fundamental niche is all the conditions where that population or species could grow and persist, but there are other factors that may prevent that population persist in some environmental condition because of interaction, for instance, with other organisms. So competition or predation can exclude some population to the environmental conditions that describe their fundamental niche. And then I will talk about geographical range, which is just the expression of this realized niche in space. So that's the set of localities where the species is actually found. And this is dependent on both the ecological requirement of the species, but also to where it actually, the kind of localities that it can reach by dispersal. So often you have this vision that you define this set of environmental conditions, which is multivariate with many different environmental factors like temperature, light, or water, and you've got this fundamental niche, which is a subspace of this space, and the realized niche will be smaller due to this interaction with other organisms than the fundamental niche. Actually, this image is too simple because where the realized niche is the result of complex interaction between dispersal, interaction with other organisms, and the fundamental requirement of the species. So this is a bit complicated, but I will just explain it to you by redrawing it somehow. So you can imagine that if you've got this multivariate space of environmental conditions, there are some subspace where actually the species can grow if it was just by itself, which is in itself some kind of theoretical idea, because maybe there's no such thing as species just by itself, but anyway. So this is where you could grow. But actually, there's part of this subspace, an environmental condition where you never can reach, because you never manage to reach those conditions. For instance, maybe you can grow in some climate, but for historical reason, you could never reach the continent where this climate is expressed. So it means that there's only part of this where actually you could colonize. And maybe there's other areas where you cannot grow and maintain a population, but you can send migrants. So you can also reach those environments where your growth rate will be negative, the population growth rate will be negative, but you can still reach them by migration. And so this is the first important definition that, well, I add another layer and then explain to you. Then, in this area where you could grow and you could reach, so here you might have some individuals, but the population is going to decline to extinction. And here you could have some individuals that could grow and establish a viable population. But actually, part of this space will also be excluded because there's competitors, for instance, or interactions with predators that is going to result in the population growth rate to be negative, but due to interaction with other organisms. So actually, the only place where the realized niche, there's this fundamental niche, and then there's a subspace where you're going to have a population that is actually growing and establishing and can be there and occupies this subspace of an environmental condition. And then you will have, so this is called a source population. And there are other environmental conditions where you might find a species because it can colonize this area. But there, the growth rate is negative, either because you are out of the fundamental niche of the species, it simply cannot establish a population and grow under in this environmental condition. Or it could if it was alone, but there are other species that are making the population growth rate negative. And those are called sick populations. So the reason why you might observe those sick population is that because there is migration from source population, putting migrants in those environments regularly, even though you cannot establish, if you would cut migration to those environments, you could not have a viable population, the population will disappear. So it means that the realized niche is not only a subspace of the fundamental niche, it can also contain also environments where actually, well, you see individuals in those populations, but they are not part of the ecological niche in the sense that they cannot persist in this environment without the help of migration from other environments. So this is all ecology, and the question now, so why people do care so much about those niche concepts? So those are old concepts in ecology, but they became even more popular recently, well, for about 20 years now, in the beginning of the 2000s, because of the development of a lot of projection of species range and how species will change with anthropogenic changes in climate or other change of sort of change such as land use changes, and with the desire that ecology and evolution will produce prediction about the state of biodiversity in the future or help management options and stuff like that, or predict invasion. And so what has been extremely popular, and you probably heard about it, is the development of those species distribution models, where what you do is that you collect spatially explicit information about a bunch of environmental variables, often climatic variables, and you contrast that to data about distribution of species, often presence and absence of species. So you could have absence or presence points, and through sophisticated statistical modeling, you actually, from the comparison of those two, produce models predicting probability of presence of the species as a function of different values of environmental variable. So that's what you describe as a niche model with a response curve saying, okay, what's the probability of presence of species depending on this variable and this variable? And then you can use this statistical model to project in other localities what is the predicted distribution of the species. And this was used, for instance, a lot to predict what climate change might do to distribution of biodiversity in the future, and try to contrast options. So this is just one example of it in Europe, where you contrast different scenarios. So this is showing a map with percentage of loss or gain in the number of species all across Europe for different scenarios. So in 2050, for different scenarios of climate change and different scenarios about migration of species. So assuming either that species have no ability to migrate, so they will stay in localities where the climate will be still favorable in the future, but they will not be able to colonize areas where they were not present before and the climate is becoming favorable. And they will go extinct in areas where the climate was previously favorable, but is becoming not favorable and is outside the niche. And this here, this scenario is assuming that every new locality where the climate is becoming favorable will be colonized instantaneously. So those two hypotheses are quite silly, but they are just extreme scenarios about what might happen. And you can see the impact of migration is on the loss of biodiversity. So those scenarios have developed a lot and they've been criticized for different reasons. And one reason why they were criticized is that they assume that this niche limits, that this fundamental niche, is a fixed property of species. So there are also other issues because they actually use the realized niche to project the fundamental niche somehow. So there's some ambiguity in there because interaction between organisms might change and everything. But also another criticism is that those niche limits are not fixed properties. I mean, there's very little properties of living beings that are completely fixed and cannot change and cannot evolve. And so you might also take an evolutionary point of view on that and wonder what are those niche evolving and how do they evolve and what are the selection pressure of those and why do they evolve fast or slowly. So so one way to look at it is to, in those models, is to say that, okay, what is setting the limits of the niche is those response curves, and those response curves can evolve. So I could parameterize those response curves and decide that those parameters are parameters that have genetic variation. I could measure that, and I could let that evolve. But this is a kind of black box still approach, where you don't know exactly why the response curve is like this, and probably there are mechanisms and underlying genes and traits behind those response curves that can evolve, and so you might prefer to have another approach. So this is an alternative approach to predict those species niche and range, which is more mechanistic. Is that, for instance, so this is just an example of a model that was developed essentially for tree species that is based on trying to predict the presence of species or not across climatic gradients based on the adequation of their phenology and their life cycle. So you all know what do you know what phenology is? Who doesn't know what phenology is? Okay. So phenology describes the timing of major events in the life cycle of organism throughout the season. So for instance, example of phenological trait will be the date at which you flower within a season, or the date at which you migrate, if you're a migrating bird, or the date at which you shed your leaves and set your birds for before winter arrives. So, and those traits are really major trait in adaptation to climatic environment, climate, especially where you've got very marked seasons. And and so in this model, you have, for instance, the timing of bud burst, the timing of flowering, the timing of fruit maturation, or leaf senescence and leaf shading in the fall, that determine the time you have to mature your fruit, for instance, or the time you have to acquire resources and produce sugars that will determine survival and growth of the plant. This is just a very, I won't go into any detail, but depending on those underlying traits, you predict survival and reproduction, and then you predict whether a species can actually successfully have a positive growth rate in terms of population in this environment or not. And actually, this model was applied to a number of North American, North and European tree species, predicting quite well their current range. And what you can see is that, for instance, if you play with those models, if you decide that if you manipulate the date of bud burst, which is when the birds open and release their leaves in the spring, and this is for oak trees, and you assume that throughout Europe, the oak open their buds at some date or later, or even later, you see the predicted distribution of population growth rate throughout Europe for those different scenarios. So you can see that depending on this phrenology, you can have oaks that are distributed in all across Europe or only in tiny parts of Europe. So those traits are a major determinant of the range, but also the niche, because the range is just an expression of the set of environmental conditions where you can grow or where you cannot grow. So you can do things with that as well. You can do the same, use this model to predict what would be the reproductive success of a tree, depending on its average bud burst date in different localities. And you actually have this kind of fitness landscape we discussed during the first week of this course, just for one trait. And you can see that the optimal value of bud burst date that maximizes this measure of fitness in Paris is different than the optimal date in Berlin and the optimal date in Zagreb. So those traits, such as phrenology, but there are many others that might determine those niche limits, are in general genetically variable. Phrenology has very, very high heritability in those tree species, which means that in general, those traits can evolve really fast. This is just one example in wild beet, where they started from a population with some distribution. So the trait that is looked at here is not directly the date of flowering, but the number of daylight hours necessary to trigger flowering. And so they started with a population where this trait was distributed like this, and then they artificially selected for earlier and earlier flowering, and after only 10 after only 10 generations of artificial selection, they obtained this distribution of this trait, which is completely different from the one they had at the beginning. And for and they estimated the heritability from this experiment, which was kind of typical heritability for a quantitative trait, around 50%, and which showed that potentially if selection on niche limits or a trait that determined niche limits was strong enough, you could have a very fast evolution of those traits. So there's no reason in principle to assume that those traits will be fixed completely. However, empirical evidence, and there's been claims that this assumption made by those ecological models that the niche is not evolving very fast is actually supported by empirical data. And in particular, there's something called niche conservatism, which describes the fact that often the niche of a species is evolving very slowly compared to other traits. For instance, if you look at some phylogeny and you look at species that are related, that diverge for not such a long time, the claim is that often you find that they have basically the same niche as their sister or ancestor species. So this is just one example that actually was very, that made a big impression on a lot of people. It's a study in Mexico, and those authors studied a large number of pairs of species that have diverged from around this isthmus in Mexico. You've got a big geographical divide here, so you've got a lot of pairs of species where you've got the two sister species on one side or on another side of this divide. And they know that those sister species have diverged from 2 to 10 million years, so quite a long time. And what they did is that they did the same kind of species distribution models that I described before. They used the distribution of one species, one of the pair of, in the pair of species on one side of the geographical divide. They model its niche, and then they use this model to predict the occurrence of the other sister species on the other side of the divide. And in most of the case, and they did the reverse. And in most of the case, they perfectly predicted where the sister species was based on the niche of its sister species, suggesting that there were no evolution of major change in the species niche, despite a very long time of separation between the two. So there are many other examples of that. And, and so this was used as an argument to say, okay, niche is not, for some reason, traits that affect the limits of the niche are not evolving very fast. And so it's reasonable to assume that it will not evolve very fast in a few decades when you are projecting climate change scenario consequences. However, of course, as any example that, yeah? Yeah. I guess for an argument of character displacement, you would, okay, yeah, you might. So maybe it's because there's a lot of speciation L patries that you see this pattern. There are other reasons I will not, discuss some of them. There are also theoretical reasons to expect niche trait to evolve with more difficulty than other traits, because it's harder. Also, evolving niche limits means evolving to have a positive growth rate where you have a negative growth rate. So you actually have a very different constraint on evolution when you take into account this demographic aspect, that you are evolving in an environment where the population is declining to extinction. And then the condition to evolve traits is completely different. So it's related to the models of evolutionary rescue that Maria mentioned at the end of last week. And we'll discuss models of niche evolution in a moment. So just to show you that there are exceptions to that. So because of the greater ability to produce phylogenies for many, many groups, and also the ability to model niche very efficiently with this kind of statistical tools that were developed during the 20 last years, you've got now many examples of that. So the idea is that in this group of species, I didn't cite it, it's a work by Margaret Evans. So she worked on this group of plants, and they have a phylogeny for this group of plants. with a well-defined taxa. And for each current species, they have distribution data, so they can fit a model for the niche of each species, and they can qualify this niche by some summary statistics. For instance, here, the mean annual temperature within the range of the species would be the trait that you look at as a measure of the niche of the species. And you can use this phylogeny and models of trait evolution along this phylogeny that have also developed a lot in recent years. So it's completely theoretical, okay? There's no fossil evidence that the niche of the ancestor was like that. But if you use the phylogeny and the trait of the current species, you can reconstruct through this modeling exercise the hypothetical value of, so here's the mean annual temperature of that species currently, and that's the sister species in the phylogeny, and you can reconstruct the evolution of the trait along the phylogeny, so you can reconstruct what was an hypothesis about the niche value of the ancestor of those two species, and so on. And you have this kind of representation through time, how the niche has diverged within this group. And what you see is that there are some taxa where actually those two species have diverged for a very long time, but their niche still resemble each other a lot. So this is one example of niche conservatism, where despite a lot of divergence, there's very little divergence for that trait. There are also other examples here, like this green clade, where there's a lot of diversity in the niche, despite, on relative term, more recent separation between those species. So what you can see is that there's a lot of heterogeneity in terms of some groups seems to have very slow evolution of the niche, some groups seems to have very fast evolution of the niche, and so far, at this scale, there's some proxy that explains this variation in the rate of niche evolution, but not many. Some people found that, for instance, species with long generation time evolve slower, different niche. There's this kind of evidence, but it's still very unclear what is the major determinant of, maybe it's very contingent to the history of this group. And so, okay, there's some fast evolution on the ground evolutionary scale, but you could argue that this is completely irrelevant when you're talking about evolution, about contemporary evolution of the niche. So there's other type of evidence that niche can evolve fast on a very short time scale that is observable by a single researcher. Right? This is one example. So for the moment, I told about climatic niche essentially, but there are different aspects of the niche, like the trophic niche. So for an insect, for instance, a very important part of where you can thrive or not is the type of plant that you eat. And so you can define the niche of a butterfly as the range of hosts it can actually eat and lays larvae on, and where its larvae could feed on. So this is one case that was described where in this butterfly, population was surveyed in the 80s, where they found that in a given location, which is called Rabbit Meadow in California, they found that the same species of butterfly would exploit two plant hosts, Collinsia and Pedicularis, with a larger population in Collinsia, but still quite large number of butterflies on Pedicularis, and with a lot of butterflies actually born on Collinsia, moving to Pedicularis to lay their eggs, and a little bit less in the other direction. So pattern of gene flow that was asymmetric, but still viable population on those two hosts. So these species could be called a generalist because it would exploit those two type of hosts. What happened in the early 90s, there was an unusual climatic event. It was a summer frost that killed all the larvae on Collinsia, but not on Pedicularis. So there was a disturbance, external disturbance on the system. And after that, so there was an extinction on Pedicularis, which is normal, but you would expect that after the disturbance, this host plant will be recolonized by the butterfly that previously exploited the two types of hosts. But it did not happen. What happened is, after that, after the extinction, there was still viable population on pedicularis, but there was no recolonization of Collinsia. There were a few butterflies migrating from this host plant to this host plant, but they never succeeded in establishing a new population on their former host plant. So on only a few years, you see a drastic reduction in the host range of the species, which was not easy to understand. So so this rabbit meadow population of butterfly was studied by Mike Singer, who is a wonderful biologist. I don't know if you've met him. This guy has been studying those butterfly populations for decades in California. And he has documented such shift in host plant use for those many, many, many years with the exquisite detail of, looking at the behavior and looking at the population size and going back every year for a field season in the same population. And this is the result of decades of work on those butterfly populations, showing that this example of very rapid shift in host plant use is not an isolated, bizarre case. So first of all, what you can see is that throughout the California, you've got a lot of variation in host use for the same species of butterfly. So first of all, the niche at the scale of the species is a bit meaningless because every population is actually having a very different niche in terms of the resource that they use. And the number of populations never change their host use significantly across the years, but a number of them did change, and often associated with extinction and recolonization as or fire changing the structure of vegetation. And so some of this change, so most of this change actually revert back to so here you had, for instance, Collinsia as the host that was most used, then back to Plantago, which was a new host plant, then extinction, then back to the ancestral host. And here you've got, again, Collinsia used, so formerly pedicularis used, some disturbance, and Collinsia started to be used, and then disappeared completely from the diet of the butterfly. So the question that we'd like to, so this is data. Now, as a theoretician, question is that, so what are the rules that govern the evolution of this ecological specialization? What do you use two hosts? What can you use only one? Why are you unable to use another host if you use another one already? So this idea of specialization being, in my mind, associated with this idea of ecological niche, the breadth of environment where you can actually thrive. And more specifically, when do we expect shift in this sourcing dynamics? So because in this example, you had a shift from a situation where Collinsia was a source population sending migrants to pedicularis, and with a very asymmetric gene flow between the two hosts, to a situation that is completely reverted, where it's pedicularis is a sink and Collinsia, pedicularis is a source and Collinsia is a sink. And then this is a shift in population dynamics, but what are the evolutionary consequences of such shift in demography? So this is a project I did when I did my postdoc a long time ago with Mark Patrick in Texas. And we described this theoretically a situation that is very similar to what Mike Singer described in his butterfly population, which we call migrational meltdown. And I will explain to you. So what I wanted to do now is go into the gory details of this model. So I will try to do as I was told, which is really walk you through the model. So I will use this particular model just to explain to you how we model gene flow and selection and interaction between the two. And so maybe you will tell me in how much detail you want to go, or I can skip some part of the derivation. But I wanted at least to write the equation with you step by step to see how, from a situation like that, you go into a model to describe it. So first, the idea that we have so one simple way to model it is to imagine two patches with different host plants. So one patch with one host plant and another patch with another host plant. But this could also be, and there's some migration, some movement between those two patches. We have a patch of host plant I and a patch of host plant J. And any given individual has some probability either to remain in its home patch where it was born, or it can migrate to a patch with a different host plant. And if you are born, and so the probability to migrate will be m dt for a very small time step. And individuals born in this patch have the same probability either to stay in their home patch or migrate to the other host plant patch. So there's no asymmetry built in the model. So that's an individual probability to move. So that's different from gene flow, which is going to depend on how many individuals move from one side to the other. Another way to see this model is, because only one patch of plant and one patch of plant is not completely satisfactory in terms of biology, it works also if you imagine that you've got many, many patches of one host plant and many patches of the other host plant mixed together in the landscape. And then the probability to move is simply the probability to migrate and not select the same host plant as where you were born, but a different host plant. Is that okay? So then we're going to imagine that there's some phenotypic trait that determines fitness in each patch of host plant. And so this phenotypic trait could be many things. Actually, Max Singer looked at many traits that are selected differently on different host plants. Some could be, the ability to detoxify the molecules produced by the plant. It could also be more subtle things, such as geotaxis. Like the butterfly can lay the eggs on top of the plant or at the bottom of the plant, and depending on the plant, it has huge consequences on the fitness. It could be also preference for that host plant. Like some butterflies will simply not lay their eggs on some host plant because they don't like it. So this is a quantitative trait, the same type as Maria mentioned, with some quantitative genetic variation. And what we assume is that the optimal value maximizing fitness on the first host plant is different from the optimal value maximizing fitness on the second host. And we assume some kind of Gaussian stabilizing selection within each patch. So, more precisely, so we're going to build a model with a continuous time, just for not really for biological reason, but for mathematical conveniency. So it means that what we're going to focus on is some underlying traits of individuals. So we're going to assume that there's some birth rate. And for simplicity, I will assume that the fecundity, the birth rate of individuals is the same, whatever their phenotype and whatever their environment. But then individuals can die with some death rate. And this death rate is going to be some function. This is some kind of mortality, intrinsic mortality, plus some extra mortality due to the fact that you've got density dependence. So if you've got a lot of larvae in the same patch, you're going to die more than if you've got few larvae in the same patch. And these are described by assuming that you've got some term that depends on. So let's see, let's call this. I'll do that later. That depends on the density of individuals in the patch I at time t. And there's another mortality term, which is due to the fact that if your phenotype is not matching the optimal phenotype, you have a higher mortality. And this term, we're going to assume that it looks like this. Quadratic function depending on the distance between your phenotype and the optimal phenotype. So actually, mortality is a function of your phenotype and the patch where you are. Sorry? There's no migration yet. Well, it is not an M, it's an N. That's density, sorry. So, yeah, okay. So the variable we're going to follow is density in patch I. Okay. And then I'm going to look at mean phenotype patch I as well. That's the two things I want to follow through time. But first, I'm going to compute the growth rate associated with the contribution to the population growth rate associated with some phenotype in the population. So that's a demography. Okay, so just to reorganize a little bit the thing. So you've seen already that the exponential population growth rate was just the birth rate minus the death rate. Okay, that's yeah. This one. So it describes the fact that if you phenotype, if for instance your degree of geotaxis is deviating from the optimal phenotype in this patch, then your mortality will be bigger. So your fitness will be smaller. So you have the lowest mortality if your phenotype is matching the optimal phenotype. So this quantity here is the optimal phenotype in patch I. Yeah, if you die more, so I'm going to rewrite it in terms of population, in terms of contribution to population growth rate. So the growth rate is all the births minus all the deaths, okay? So this is all the births minus all the deaths. And so you can write it like that. Oops. And just to rearrange things a little bit, I'm going to call big K the number of individuals in the patch at equilibrium if no migration And if all individuals with optimal phenotype okay, that would be if you cut migration and you let the population evolve and you assume that all individuals have the optimal phenotype, there's no genetic variation anymore, then you will get this number of individuals in this patch. So this is a measure of the carrying capacity of the patch, so how much the patch can sustain in terms of individuals that would be perfectly adapted. And so you can rewrite this R if you do that, and you can call this quantity here R0. R0 would be the growth rate at low density of individuals with optimal phenotype. You get this expression for the growth rate of some individuals with phenotype Z at time t in patch I. Okay? So, should I try this? So okay, so I can define another parameter that we might need. So this parameter gamma here, so if gamma is big, you've got more mortality when you deviate from the optimal phenotype. Okay? This mortality term will be bigger if gamma is large. So gamma measure the strength of stabilizing selection, which is bringing you near the optimal phenotype. Gamma is large, means that if you deviate from this optimal phenotype, you have very low survival. If gamma is small, it means that you can do well, even if you're not perfectly matching the optimal phenotype. Okay, so then the last parameter I told about already, it's a rate of movement M. We just described that the probability that you change host plant. Yes? No, the B, no, we assume not. We could, it would not change much. So I could write everything assuming all selection is on mortality and not on birth rate, but you could actually do it the other way. It's just the same, because in the end, if you look at this equation, I'm not going to use that anymore. I'm going to use this. And so, you've got this R naught, and you've got this term here. You could imagine this term is coming from the fact that the birth rate is declining. So you could, do both. There are cases where it would matter, but not in this model. So the rate of movement is just the probability that an individual moves between the two habitat patch between a small interval of time dt. So So I say there are two variables that I want to follow in time, which is density in each patch of host plant and the mean phenotype in each patch of host plant. So first I'll try by change in density. So I look at very small time. I'm going to look at what's going on when I go from time t to time t plus dt. Then I'm going to have a very small change in the density. So could you tell me what I should write there? What would result in a change in population size in this small interval? So what if I change the number of individuals? What should I count first? Yeah, so how much? So I can say I want to have the change will be all the new individuals, the births, minus all the deaths, and then I want to put in all the immigrants and remove all the immigrants. And the fact that I'm adding all those is because I'm looking at a very small interval, so I assume that any individual can do only one thing. It can give birth, die, emigrate or immigrate, but they cannot do several things. Okay? So if I was, so I start with migration only. Okay? For instance, for the moment, I will ignore that, and I will compute only the change in population number due to migration. So how many immigrants do I expect? So here I've got NI individuals, here I've got NJ individuals. So in a very small time dt, how many immigrants will I get? M NJT dt. Okay, that's those that come in, and I have to remove the same term for those that come out. That's very simple, which means that I will remove the T notation to simplify things. But all right, now, if I want to do the change in growth, so one thing to note here is that the change in population density just depends on the difference in density between the two patches. So if you've got the same density in the two patches, there's no influence so far, no influence at all of migration on the number of individuals you get in the two patches. Okay? No direct influence. So if I want to compute now how the change in density in the patch due to growth, what I need to do is I need to compute the average growth rate in patch I of individuals, okay? Because I've got individuals with different phenotypes, they don't have all the same growth rate. So if I want to know how many individuals I've got in total, I have to integrate across the distribution of phenotypes in that population. So I have to compute this mean population growth rate at time t. So again, I will assume a continuous distribution of the phenotype in the population. And so I will integrate phenotypic values, which is minus infinity and plus infinity. And for each, I've got some growth rates that depends on the population size and the phenotype of the individuals. And then it depends on the frequency of individuals with phenotype Z in the population. So so if you do that, in this if you use this equation that I circled in yellow and replace it here, you can then find that this quantity will be equal to this. So here we've got a new term here, which is the phenotypic variance for trait Z in the population in the patch. And this here is the mean phenotype in the patch. So you see that the population growth rate is depending on the population density through regular kind of logistic density dependence. That is depressed compared to the population growth rate you would have if you had the phenotypic, if every individual had the phenotypic phenotypic value equal to the optimal value by two types of what we call genetic loads. One is called the variance load. And this other negative term, which is depressing the mean population growth rate, is called evolutionary load or lag load. And Maria also mentioned that on Friday. So what do those terms tell you? That so if you mean phenotype is displaced from the optimal phenotype, then it's quite intuitive that the population growth rate will be smaller than the maximal population growth rate you could have at this density, because then your population is maladapted, it's shifted away from the optimal value. But even though the mean phenotype would be matching the optimal phenotype, so the mean of the population would be at the optimum, but you've got some variance in phenotype in that population. So not all individuals have the same phenotypic value, which is generally the case in every population you look at. You then have also a depressed fitness or population growth rate due to this variation in the population, because simply individuals deviate from the optimal phenotype. So the more variance you've got in the population, the lower the mean fitness is. So this is sometimes a bit counterintuitive, because phenotypic variance is seen as something that, or genetic variance is seen as something that allows adaptation and should improve fitness. But once you reach some optimal value for fitness in terms of mean population, then the remaining variation that you have is actually depressing your mean fitness, and that has some impact on the demography. Okay, so I could, do you want me to derive that, or is it okay for you to assume that it's true? Or you want to know how you get there? I don't know. It's a bit boring, but sometimes there are some results like that you're frustrated not to have the proof ever, but so what do you want to do? Proof, no proof? Okay, no proof. So if we add up those two things, we find that, yeah. So here I forgot a little dt. So the rate of change in population size in patch I is then quite simply so let's turn to the mean phenotype now. Okay, so we'll define change in the mean phenotype through a very small time step. And this mean phenotype will change about because of two evolutionary forces in this model, which is quite simple, which are what might make the mean phenotype change. Sorry? I didn't hear. What is producing evolutionary change in general? Mutation, okay, I will not consider that. Okay, something I need to say. Yeah, I forgot to tell you a lot of things. So, what I will assume, okay, mutation is causing change, but this I will somehow hide behind the carpet for the moment. I will explain to you how. So what is producing change in mean phenotype as well? Selection, so this we're going to do, and migration. So we're going to do selection, migration, and mutation will be somehow neglected at the scale where we can look at, or not explicitly model. So what I'm going to use to describe change due to selection is the same kind of Breeder's equation that Maria introduced to you last week, assuming in this version of the model a constant genetic variance, the same genetic variance in both patches of habitat, and a variance that I will consider as a parameter, and that will not change during this small time step. And I will also assume to describe the effect of selection in this case that in each patch, breeding values for phenotype are distributed as a Gaussian, so, which is not such a bad approximation when you have many, many loci affecting the trait, okay? But both those assumptions are likely to be a problem, actually, when you've got migration in some heterogeneous environment. So we're going to write equation like this and see what the model says. But what we did in the paper is that we also checked that we didn't get it too wrong by making those assumptions that are likely to be not true, by actually comparing that to simulation with an explicit genetic architecture for the trait, where the genetic variance will evolve and where we've not assumed the distribution of phenotype to be Gaussian necessarily. I'll show you that later. So if we look at the change in mean phenotype that we would expect under this assumption due to selection, what we will, if I just use a version of what Maria showed to you on Friday, or before, I don't know. Right. So I will use a version of the Breeder's equation where the change in mean phenotype through a small time step would be equal to, that's a genetic variance additive for trait Z. And this is what Maria called a selection gradient. And what she defined as a selection gradient on Friday was a covariance between tree Z, and what she used in her notation was the relative fitness, so the fitness of an individual with tree Z over the mean fitness, everything standardized by the phenotypic variance. So actually, if we assume that the distribution of Z in the patch is Gaussian, you can show that this gradient can also be defined as the derivative of the log mean fitness by the mean phenotype. So this is in a discrete time model, and the version of that in a continuous time model is that the selection gradient is equal to the derivative of the mean fitness with respect to the mean phenotype. So you can go from there to there quite easily by computing this covariance and assuming that this is Gaussian. If you want me to do it, to tell me. I can also show that. Okay. No migration. Should I? Oh no, maybe we should finish selection first. Yeah. So unfortunately, I erased that. But so if we compute this, in all case, given it's in there, given the expression that we have here for the mean fitness of the population, if we take the derivative of this with respect to this mean phenotype, quite straightforward to compute, and then you have an expression for the selection gradient, which means that you find that this is equal to gamma So the more you depart from the optimal phenotype, the stronger the selection gradient. So the stronger selection will push you back towards the optimal phenotype. Migration now. So let's assume we had migration only, and then we had all the terms. If I want to compute the mean phenotype after migration, how am I going to do that? So I had some mean phenotype in patch I, and patch J, and then there's been some migration. And if I want to compute again the mean phenotype, what do I need to do? I'm just computing the average phenotype in the patch, okay? So I had two patches, I mix them, I have to compute again the mean phenotype in this mixture. So in this mixture, how many immigrants do I have? No, how many residents do you have? How many individuals stayed in the patch where they were born? So they were initially NIT individuals, but how many of those stayed? One minus M dt. Okay? That's all those that went away, and so I've got all of them minus those that went away. And those had what kind of phenotype? They had ZIT. Okay? Then I've got all the newcomers. And how many newcomers do I have? M dt and the size of the other patch, J. And those had mean phenotype J. Okay, that's just, I have to rescale this, okay? Because I'm computing the average. So then I have to divide by the total number of new individuals after migration, which is just so now what I want to do is I want to actually compute this dz over dt. That would be that quantity that we just computed minus what we had before divided by dt. And what I want to do is take a limit of that when dt is very small. Okay? And if I do that, I find that the change only due to migration will be proportional to the movement rate, the ratio of population size, and the difference in mean phenotype between the two patches. So the effect of migration on phenotypic change is depending on the difference in phenotype between the two environments, but the ratio of population size. And what is important here is that it connects. So, well, I'll show you later. So you've got demography that is mediating the effect of migration. Actually, the ratio of population size is as if it was changing the migration rate, okay? And affecting this. So, for instance, what you can see from that is that if you have, for instance, NJ being much bigger than NI, so if you got one patch much bigger than the other one, then you will have a very strong effect of gene flow on change in mean phenotype in patch I, okay? Because this will be big. Contrarily, if you've got Ni bigger than Aj, you'll have almost no effect of gene flow on phenotypic change in the population. The relative population size is determining the impact of gene flow on the evolution of mean phenotype. Okay, so we have two equations. So, okay, so if I sum up those two terms, I want to have change in mean phenotype through time. Then I have, Then I have a term depending on selection. Yeah. Yeah. So, for instance, it simply means that, for instance, if the patch sending immigrants to you has a small value of the trait, the effect of migration will be to decrease the value of the trait in your population, because the term will be. If just an example, if this is smaller than that, so if, for instance, I've got big butterflies and small butterflies, and I'm in a patch with small butterflies, and I'm receiving migrants from a patch with big butterflies, so that J is greater than that I, then this will be, so if this is greater than that, it means that the change due to migration, I mean phenotype, will be positive. So I will increase in size because I receive migrants that are bigger than me. So that's, and the reverse, okay? Yeah. So that's a term for selection. And that's a term for migration. So we've got those two equations, well, actually four, because we've got the same equation for the other patch. We've got a system of four equations that describe the joint dynamics in evolution of the trait and population size in the two patches. So because in general, theoreticians like to reduce the number of parameters, I'm going to rescale this equation. So you've got parameters that integrate several parameters that are easier to manipulate, but it's good to derive them from first principles to see where they come from. So if I do this rescaling, so I will rescale in okay, a new quantity, which is not directly mean phenotype, but I will call maladaptation. So this quantity is defined as, so I will assume in the following that the optimal value in the second patch is greater than the optimal value in the first patch. It's just it's completely arbitrary, okay? And I will define the maladaptation as the distance in terms of standard genetic deviation between the optima and the mean scientific value in that patch. So if this quantity is greater, it means you're lying further away from your optimal value, and this is rescaled, so there's no unit because this genetic standard deviation is in units of phenotype, and this is in units of phenotype, and this is just an n-scaled measure. So similarly, I can do the same thing for the other one, and I will define it so that it's also positive. So my adaptation is always positive. The other thing that I will do is I will scale the population size. So I will call big NE. The population density scaled by a carrying capacity, but this carrying capacity is the equilibrium population size in the absence of migration, which I have here. Oh, well, first of all, I'm going to define another quantity, which is R star. Which is the maximal population growth rate, taking into account the fact that you've got this variance load, so you cannot get rid of the phenotypic variance in the population. And the carrying capacity K star that we'll define is scaled by this ratio of population growth rate. So that's actually, this is the carrying capacity in the absence of migration, but taking into account this variance load. Now, the other scaling that I will do is that I measure now things in units of time that are different. So instead of measuring in standard unit of time, I will measure time by multiplying real time by this growth rate, which means that I measure time in numbers of generation and not in standard unit times. And so every quantity that depends on time, it will now be scaled by this new time unit. So M, big M is defined this. And then I have the strength of selection, which is the product of the genetic variance, the strength of selection, also scaled by my new time unit. And finally, I will define another quantity, which I call H, which is defined as this. Which is also unitless and depends on the difference between the optimal value in the two patches. So it's a measure of habitat heterogeneity. So it's a measure of how different is selection across the landscape. So if I do this, I get a new set of equations. Before we go for lunch, I just want to, so that's what we already saw. So those two sets of equations show how the feedback between adaptation and demography work in this model. So change in population growth rate are coupled to change in mean phenotype because of the population growth rate, depending on the degree of maladaptation. So if the maladaptation is big, population growth rate is smaller, and so the rate of change in population size is smaller. So that's one way the two things are linked. And the reason why in this model evolution is linked to population dynamics is because the relative population size here, so the size of one patch respective to another, affects the effect of migration on the change in mean phenotype. So the result of that is that, so if you have a population that is very small, so if this is small with respect to that, this term is going to be very big, and it will, you see that this term is positive, so it increases maladaptation. So if maladaptation is increased, then this term is bigger, which means that population size is smaller. And if population size is smaller, this term here is larger, and maladaptation is bigger. So you have this kind of feedback loop, positive feedback loops, where if you start with a population with a smaller relative density, it suffers from increased gene flow because then the relative population size matters, and which generates increased maladaptation, which decreases again the population density. And tomorrow we'll see what kind of interesting behavior this can generate because of this locked-in feedbacks between demography and adaptation. If you have questions, I can take them now or later. Okay, you're all hungry and tired with mathematics? Okay, let's go for lunch.